All right. So a couple questions first. Pectin lab, yeah. Okay. So this week on Friday, to answer some questions that people have, the questions for Common Grounds, those questions that I had given you need to be submitted through this portal. So that was the questions that were inherent in that lab protocol, the Common Grounds 3 protocol, and then like the charts that I had asked you. I realized some of those things are not going to be highly correlated, that I asked you to look for a correlation. Couple things. First of all, that's the way science goes. Sometimes when you're looking for correlations, you don't find them. Did I know whether there was going to be a correlation between the size of apple seeds or the number of apple seeds and the size of apples? No, I didn't, but I was guessing. And we didn't find anything there. That's okay. Report that. That's the null finding is significant in and of itself. So that is something to report. Next year, we won't look for that. We'll look for other correlations. There are some correlations that exist in that data. The other aspect of that is that, in fact, not all of the calculations that were done in the data spreadsheet were done correctly by everybody, and that's something that those individuals will be getting graded on. QA, QC, quality assurance, quality control of a data set is an important part of science. And so when you're going through that spreadsheet and you're plotting some of that data and you're like, wow, that point is crazy out of line with the rest of these, you look at the data and you see how that number was calculated and say, okay, wow, that wasn't done right. That's what QA, QC is. That's an important part of all scientific data reporting. So those are the two things that you should keep in mind as you're making the plots for the correlations. The physical properties of food, the pectin, and so that had been due last week, as I had told you in lab 2 weeks ago when we did that lab, but I extended it because a few people had some things with the rollout of the capital campaign, and I don't know, a number of people asked for an extension. That's totally fine. I gave you an extension. I'm not like keeping a strict eye on it, but I would like those in just to keep your workflow going by Friday. The other one that's due on Friday that I'd hoped you would write up is the physical properties of food, the pectin write-up. There was a few questions in there. Please answer those and submit those in a reasonably timely manner. Here's the lab for this week. I spent a good amount of time writing this up. The questions are very clear in there, and we'll do the lab on Friday, and then that will be submitted. The answers to those questions will be submitted the following week. Should be pretty straightforward. The lab on Friday, I was hoping to actually have started lipids by this point. We're still in carbohydrates in the lecture, but the emulsions lab is a nice transition between carbohydrates and lipids. You'll see that as you read the lab and understand what that really means. All right. So yeah, in this thing we are right here, physical properties of food. So one of the other things I want to do on Friday is take a sample of our scobies, pack those off, and get them in the mail so that we can do the isotopic analysis for lab down the road and see how that experiment we ran turned out. So that'll be all on Friday. I'd like to get out of there, get you guys out of lab by 3 o'clock, hopefully, if you guys are working in teams efficiently. I think it should be no problem to be done by 3 on Friday. Any questions about any of that? Ask now. Okay. Last time I ended by talking about how sugar used to be made from sugar cane, the process that sugar was, how sugar was produced. And I'm going to skip the rest of that. I don't need to go over photos of obscure sugar factories any more than I already have, except to remind you that there is a grim history in many of the chapters in food science. So I launched into that discussion by talking about two common disaccharides, sucrose and lactose. And sucrose led us to the discussion of how sucrose was produced. Hopefully you've listened to some of those podcasts that expand upon that a little bit. We're going to switch gears to consider lactose now, the sugar that is principally found in milk. I once again have a lovely apple right here, and I'll tell you, so these are the stakes. I'll tell you how you can claim this delicious apple from one of my trees in my yard. No worms, nothing but delicious white Macintosh apple for you to chomp on if you can get it. But part of it is related to lactose and these molecules that are sitting in the middle of the room over here. So this sugar, lactose, is made of a galactose sugar, which is over here on the left in this diagram. And we know it's galactose because it looks like glucose otherwise, except for the fact that it has this one axial hydroxyl. In glucose, this should be equatorial. So this is a glucose ring, and we see how it's equatorial here. So the stereochemistry around carbon 1, 2, 3, and 4 is different between glucose and galactose. Do you want to see one of those 3D models of galactose again, or lactose, to refresh your memory? Or is that, you want to check that out? All right. Here it is. Okay. So here is the non-reducing end, because here's carbon 1 that is attached to 2 oxygens and is part of the glycoside. And this is the reducing end. Here's the anomeric carbon on the glucose. Reducing because this hydroxyl group's not attached to anything, and this ring over here can open and close at will. So let's look at the non-reducing end, and we'll flip it around. And we notice, so we've brought the plane of this sugar, our eyes into the plane of this sugar ring. We see this hydroxyl is sticking up. That is the C4 galacto. If we were to look down the center of that ring right here, we'd see that this is indeed an axial hydroxyl. It's pointing right towards us along the axis of that sugar ring. And on the other side, we have glucose. Here's C1, 2, 3, 4. This is equatorial. It's not pointing at us when we're looking down the axis of the ring. And when we look in the plane of the ring, we see that it's also laying in the plane of the ring. And instead, it has a hydrogen sticking up axially. All right, so that's the difference between glucose and galactose. That's it. To make lactose, milk sugar, the glucose and the galactose are linked by this beta 1,4 linkage. Okay? 1 because it's coming from the 1 carbon in galactose, and 4 because it's going to the 4 carbon in sucrose, or in glucose. All right, back to it. So both galactose and glucose are really useful sugars for the body to use. And when we eat lactose, when babies drink lactose, they produce an enzyme that is called beta-galactosidase down here, or lactase is a more common name for it, lactase. Beta-galactoside, a beta-galactoside is another name for the type of linkage. So it's beta-galacto refers to galactose, a beta-galactoside, glucoside, glycosidic linkage, all right? So beta-galactosidase is an enzyme that's going to cleave a gal beta 1,4 linkage. But they also call it lactase because that linkage is found in lactose. All right. this is the enzyme. And this is how it works, much like the sucrase that we saw last time for sucrose. Lactose comes, it binds into the active site of the lactase enzyme, and water joins the party and hydrolyzes that glycosidic bond, that ether linkage that is going across the hemiacetal here, that glycoside, that linking, bridging oxygen. That bond gets cleaved and water adds across that bond, liberating both the galactose and the glucose. All right? And then those sugars are released from the enzyme, and that lactase enzyme has the active sites open and free once again to go back around this catalytic cycle. Lactase is not consumed in the process. It's just facilitating the process, lowering the energy barrier like we talked about when we first introduced enzymes. Yeah, Damon.
So, like, that's just a visualization of what happens.
Yep.
Lactose, but like after the lactose has been hydrolyzed, like where does the galactose and the glucose go?
Yeah, we'll get to that. Trust me. So they go into your bloodstream, into the nutrient pool, and that's probably actually happening in a cell, perhaps, and those sugars are going to be utilized for other things. So the glucose can be used to burn. You can burn it and make ATP and energy out of it, or you can assimilate it into other anabolic pathways. It can be incorporated into larger structures if you need it. And galactose is probably going to be assimilated, anabolically assimilated. We'll see that. We're not going to, I mean, you know, I'm trying to open the door a little bit so you can see the bright light shining through the crack, but I'm not going to like rip it wide open. We're on the verge of an enormous world, I think you can probably appreciate. But the galactose and the glucose are both going to be used for a host of different things in the body. All right? Does that make sense? We could, if we had a real need for glucose, you could turn the galactose into glucose just by epimerizing. It's called epimerization, where you would flip the stereochemistry of that carbon at C4, and so the galactose would turn into a glucose.
You can't do that by yourself, right?
You have an enzyme. There's an enzyme that does it. Yeah, they're called epimerases. It's an enzyme that'll do that. Okay? Good. That was a good question. Anyone else? Okay.
Can you talk about lactose intolerance?
Yes, I can. So, turns out that there are certain people after a certain. How do I start out talking about it? Infants have beta-galactosidase. They make beta-galactosidase. At a certain point, primates or mammals in general, stop drinking milk. They go from infants, whatever that mammalian infant is, suckling and nursing, and then they are weaned off of the milk. They lose their milk teeth, and they gain their adult teeth. And the other thing they lose is the production of beta-galactosidase. They stop making lactase. So there are many humans that do not make beta-galactosidase. And this makes it difficult for them, or impossible in some instances, for them, many instances, to assimilate and break down lactose. It wasn't a problem. Before the domestication of cows and goats and sheep, was it? Because the milk, the only access to milk that we had was mother's milk, and that was really just for infants. Mothers would dry off after the infant was weaned. Okay? So, however, the question is not where does lactose intolerance come from, but where does the persistence of lactose tolerance the persistence of lactose tolerance come from. So people who can still drink milk as adults. So apparently, the mutation that happened in humans sometime after the advent of agriculture and husbandry happened in Europe. And this makes sense because this is one of the first places that the common production of cow milk for consumption by humans was adopted. Right? And they say that about 9,000 years ago, there was a mutation in the population somewhere in Europe, and that gene spread throughout Europe and across much of the planet because it turned out, based upon the dietary preferences, the changes in dietary preferences, and the ascendancy of domesticated dairy cows in the human diet, that this gene for the persistence of beta-galactosidase into adulthood was an advantage. If you were able to drink dairy milk, it was an advantage to you. It became an advantage. And so the human population, currently we're in the midst of the human population converting the overall gene pool. All right? There are some people who are lactose intolerant and some people who are lactose tolerant still, and you have the persistence of beta-galactosidase. That conversion, the adoption of adult beta-galactosidase, is not complete in the human gene pool. There are many portions of the world that they don't actually drink very much milk. Okay, so this cool GIF that I have up here shows a computer simulation from some researchers that in the UK. You can see the reference down there, where they did a broad genetic sampling across people of Northern Europe, and they traced back the initial site of where the genetic mutation happened, and they correlated that with migratory information and genetic cross-correlations that helped map migratory patterns of humans in Europe over the last 9,000 years. And this is a model simulation of the spread of the gene for the persistence of adult beta-galactosidase. So this is, in the beginning, when you see it's all blue, everybody was lactose intolerant 9,000 years ago in Europe. And then where you see the X, you see that X somewhere in central Germany, that's apparently where the initial mutation arose. But then you see that with the Saxon invasion of England, that gene, so initially, let's watch here, we see it first begin to start and spread across Northern Europe, but then there was the Saxon invasion of England, and those genes spread to the UK and Ireland rapidly because those were very good places to have sheep and goats and even dairy cows. So the consumption of dairy in England and Ireland was a significant evolutionary force, preferring the prevalence of lactose tolerance in adults. You guys follow the story here? So this is it. The baseline is not lactose tolerant. It's lactose intolerant, and the population is shifting towards adult lactose tolerance as dairy becomes a larger and larger portion of the human diet. This took about 10,000 years for this to happen. All right, so essentially since the last ice age. As soon as the last ice age receded and animal husbandry came to the fore, that's when the advent of the persistence of beta-galactosidase or lactase happened. So people who still haven't had their gene pool mixed in with this gene in the human gene pool that is adult persistence of lactase, they can still eat dairy. And the way they do that, there's this, is anyone here lactose intolerant? I am not, actually. Okay, so, right. And I'm not exactly sure what your ethnic background is, but it tends to be a lot of Asians are lactose intolerant, and that's because if you look at what I'm showing you here, you can begin to appreciate why that is. The persistence of lactose tolerance started in Europe, and those genes haven't migrated fully to the gene pool in Asia currently. So, but people who are lactose intolerant can take this stuff called Lactase. Have you ever tried that, Nick? You ever taken that stuff? Does it work?
Yeah.
Yeah. Okay. Interesting. So what is it? It's just. Does anyone want to guess before I just tell you? What do you think it is, Hannah? It's lactase. It's the beta-galactosidase enzyme in a convenient lyophilized pill form, or maybe it's powder. I've never taken it. I don't know. I'm happy with milk. Yeah.
So I was lactose intolerant when I was like 10 to 12. But then we thought I had neurotics and not anymore. Does that make sense?
Yeah. So there is there are first of all hormonal components to that, and perhaps you have only one allele for it. Maybe is one of your parents lactose intolerant, or maybe recessive. Maybe they also only have one allele, but you got lucky and got the intolerant allele from them, the gene that got passed down. So, and that can be hormonally influenced. So a lot of times that's correct. People will grow out of their lactose intolerance, or they can grow into their lactose intolerance. It's guided by a lot of environmental factors. This is a matter of nature versus nurture. Does that make sense? Yeah. Okay. Anyone else? Questions? Yeah.
So why does does do milks like soy milk and almond milk, do they just not have lactose?
Totally. Yeah.
Is it only in like animal milk?
Yeah. Yeah. It was meant to be. So, I mean, soy milk and almond milk, it's not like you go to the almond tree and there are mammary glands and you milk an almond tree and get a bucket of it and, pour yourself a frosty glass. They take almonds, they soak them, right, for a long period of time, and then they put them in a blender and homogenize it and press it and essentially extract all of those proteins that have been, and fats and whatever, that had been liberated from the soaked and masticated. So you would use something like that juicer you guys used on Friday, a masticating juicer. You could actually make almond milk with that thing too. You just soak them, you hull them, you take the skins off, you soak them, the white almonds, and then you pour the juice and the almonds through the thing. It blends up and it suspends all the stuff, and you'd have your almond milk. Yeah, but there's definitely not lactose in there. There's not lactose in there. Which is why people like yourself can drink almond milk and soy milk and any other kind of non-animal milk without having a problem, because there's no lactose. Any other questions? These are great questions. Ask me more. This is what I want. Questions? Yeah. That would be a problem. There are infants that are lactose intolerant, but you have to understand that being lactose intolerant as an infant is never really an advantage. Being lactose intolerant as a human, an adult, wasn't the advantage to it was that you weren't drinking milk, and why put all this energy into making a protein that you're not using, right? So you save energy and resources by becoming lactose intolerant before the advent of dairy. But for infants, it is never an advantage because that's their primary source of food. And I'm about to continue the story of lactose here and explain why. But Hannah's question comes first.
When you answered Anna's question, you were talking about like environmental influences.
Mm-hmm.
Can you like elaborate on what that would entail?
Yeah, so I was talking about, again, I'm going to give you a very superficial answer on a topic that's extremely complex, and my understanding is more than I'm going to give you, but my understanding is also incomplete to give you the complete breadth of that, because we're talking about sort of generalities anyways, and so there are specific case-by-case considerations. But essentially, so for example, a person who has asthma, and I'll talk about asthma for a moment. There's been a treatment for asthma where where they expose people to hookworm, and it's these kind of repulsive worms that you pick up by walking barefoot in a latrine, in like feces, and they go through your bloodstream, they get in through your skin, go in through your bloodstream, and lodge themselves in your lungs. Kind of gross, but it turns out that if you have hookworm, if you get hookworm, your asthma goes into remission. And without getting too in-depth into explaining why your immune system works this way, I'll say that asthma happens because it's an autoimmune reaction by parts of your immune system that were designed to deal with parasites, but because we all live in such a happy, shiny world over here in North America, none of us get hookworm or any other kind of parasite. I mean, who here's had a parasite? So the parts of our immune system that were an evolutionary advantage for millennia, for eons, because humans are animals and came in contact with parasites, are not being used now, right? They're not being used. And so those parts of the immune system don't learn from their environment. Like your physiology is kind of heuristic. You know what that word means? Heuristic means learns from experience, learns from its context and experience, right? So by changing the experience that the asthmatic is having by giving them hookworm, that part of their immune system, which has been causing asthma, attacking the lining of the lung, says, oh, this isn't what I'm supposed to be doing. I'm supposed to be doing this, getting rid of the hookworms. Okay, and it starts doing that. So that's what I'm talking about in terms of environmental cues. And to give you a more specific answer, you'd have to give me a more specific case, right? But in general, there's all kinds of things that can happen to a person that can lead to their stress or lack thereof, that can lead to pro-inflammatory or anti-inflammatory cytokines and hormones and interleukins and leukotriene chemicals in their body that are going to result in different manifestations of your health, right? So you can take your body down different paths. You have this it's like having a set of tools, right? With that set of tools, you can build a house, or you can build some cabinets, or you can build like some Christmas toys, or you can build like a tree fort. Any of those things, it's just what direction you're going to take your biology in by the context of your life. Maybe you go and get a really bad case of Epstein-Barr or mono or something like that, and it does something to your immune system, and it slightly changes the trajectory of your life. Who knows what happened to her when she was 10 years old? I mean, she was about to go through puberty, so that was certainly one of the things that was happening for her. So, there's context affects your biology for sure, and that's the whole notion of nature versus nurture, right? So nature are the genes that you're born with, and nurture is the context that changes that changes the outcome of your biology. Does that make some sense to you? All right. It's just like, you can think in very simplistic terms. You can take a set of twins and you can stick one of them in a broken home where the single parent works 3 jobs, one of the parents is incarcerated, and the brothers and sisters are abusive or drug dealers or users or something like that, right? And you can take the other twin and put it in like a supportive, household where they have all the resources they need. There's never any like anger problems. They're being read to at night. They're going to the park and the library and having all the food they need on a daily basis to be healthy. You know, and they're not under constant stress. Those two, I mean, there's going to be an outcome in what happens, who those adults become, and potentially that can manifest itself in their health, right? And in various ways. It's like a kind of a simplistic metaphor for you there. But is that overkill? Okay, sorry. It's a good question. It's important to think about. Anything else before we talk? Yeah.
Do animals have this thing where when they get older they have this lactose? They don't make it anymore? Like,
The animals.
I think about a cat drinking a bowl of milk or something.
Yeah. I don't really know a lot about lactose intolerance or persistence in other animals, frankly. Cats, I don't know. Yeah, cats drink milk. Do they feel good afterwards? Does any cat live exclusively off of milk, or is it just kitties that are drinking milk? I don't know. I'm allergic to cats. I stay away. Can't answer about cats. But that's a great question for you to research. Tell me about it. I'll learn from you. Anything else? Okay. So, lactose. First of all, when you drink, when a mother, so we see here a mother nursing her infant. She's producing milk, and milk is a really complex combination of different nutrients that we probably won't have a time to talk about in detail, but we'll talk about some of them. And one of the major components is lactose, of course. So you may think, wow, what is that lactose doing? What is it doing? Well, the baby's absorbing it. I just talked about lactase. The baby's clearly absorbing that lactose, breaking it down into glucose and galactose, and growing into a strong young child. That's partially what happens. In fact, the bulk of the lactose that the baby is nursing, is consuming, is not actually absorbed by the baby. In fact, the bulk of it is being absorbed and assimilated by the bacteria in the baby's gut that the baby was inoculated with when it was when it came down the birth canal. We talked about that in the beginning of one of the Sander Katz podcasts. We talked about infants getting inoculated by the female perineum after it emerges through the birth canal. Those bacteria, particularly E. coli, inoculate the baby, goes into their intestines and populates their intestines. It's those E. coli that are actually absorbing a lot of the lactose. E. coli they're prokaryotes. They're bacteria. They're not eukaryotes like us. Eukaryotic cells, I'll remind you, have nuclei in them that has all that DNA. Prokaryotes don't. They don't have a nucleus. They just have strands of DNA in their cell as they are. Okay? And this DNA, this bacterial DNA, is broken up into operational units. Operational units called operons. An operon is a set of genes. The gene products of which, the proteins that are made by them, the gene products of which have a concerted function, a unified function. So, for example, what has been called the lac operon is an operon in E. coli. The overall function of the lac operon is to absorb and break down lactose. All right? So there's a number of proteins in the lac operon. Here in this diagram across the top, you see the lac operon. It's a strip of DNA that's found in the bacteria in the gut of a baby, the E. coli, Escherichia coli. There is the lac Z, lac Y, lac A, these different proteins. One of the proteins is a beta-galactosidase, the protein we just talked about that breaks down lactose into galactose and glucose. There's a permease that gets it in. That's an enzyme that gets the sugar in the cell, in the bacteria. There's a transacetylase, which we don't need to worry about what that is. The important one is the beta-galactosidase. All right. So, however, bacteria, E. coli, aren't just going to, the word is constitutively, produce beta-galactosidase. It's not going to constitutively turn on the lac operon. Constitutively means all the time. A constituent is someone who's there all the time representing you, or is a member of a unit. So constitutively is something that is constantly on. Something that's constitutively expressed is on. It's not. It wants to turn it on when there is food around. It needs a molecular cue to tell it when to turn on the lac operon. It seems like lactose may in fact be a good effector or key to turn on the engine, but it's not, because lactose itself is the thing that is getting consumed by this reaction. So there's another molecule in there, and we call it allolactose. Allolactose is just like lactose. It's galactose beta-linked to glucose. However, rather than being beta-1,4, as you see in lactose over here on this diagram, here is lactose with the gal residue over here and the gluc residue over here, beta-1,4. But over here is the allolactose. It is gal because here is the axial O4, that's a gal, but it's beta-linked, because this is equatorial, to the 6-hydroxyl, the carbon 6, this big hydroxymethyl tail that sticks off the side of the sugar ring. So this is a beta-1,6 linkage, allolactose. Allolactose is extremely trace in milk. There's only a tiny bit of it in milk, but it is essential to be able to turn on the lac operon. Bacteria cannot absorb mother's milk without allolactose being there. This is one of the numerous reasons why, if it is possible for a mother to nurse their infant, they should. Because allolactose is extremely difficult to synthesize. I can attest to that. I'll show you a slide in a minute. I spent about 7 years of my life trying to make the stuff with C13 label in it and crystallize it. It's extremely hard to make, so they don't put it in formula. It's not in baby formula. All right. So the lactose that's in baby formula, babies are eating it, but the bacteria are not in their guts because they don't have the allolactose to turn on the lac operon. We'll talk about why that's a problem in a moment. All right. So when allolactose is present, it turns on the lac operon. Turns it on so that the bacteria can then make beta-galactosidase, which helps the bacteria, the permease, which helps absorb the sugar, and the beta-galactosidase or lactase, which helps break it down into the gal and the gluc. You guys following that? So this is real biochemistry of food here, starting at the beginning, babies. Let's keep going. So what's interesting about the lac operon is that it was the first example of genetic control that was ever identified. They didn't really understand, previous to the elucidation of the lac operon in E. coli bacteria, researchers didn't really understand how genes were regulated. These guys figured it out in the late 50s and early 60s, which is kind of incredible considering we didn't even know the structure of DNA until maybe a decade before that with Rosalind Franklin, and Watson and Crick. Well, anyways, these three guys, French dudes, Francis Jacob, Jacques Monod, and Andre Wolff. I don't know how to say that last name. Sorry. They got the Nobel Prize in 1965 for identifying allolactose as being the effector of the lac operon, the thing that turns the lac operon on. I'll just say that these two guys lived ripe old ages. Unfortunately, this rather dashing man, Jacques Monod, died quite young in 1976 because he was a smoker. Please don't smoke. I know some of you may, but try to stop while you're young. It's bad for you. All right, so here's the reason that the lac operon is important. So here's another mother who has been nursing her baby, and this baby has dumped a load in its diaper. Rather than, I contemplated for this slide showing you guys the picture of a dirty newborn baby's diaper. Has anybody ever seen one? Anyone have like brothers and sisters you had to change it? What does it look like? Just tell us.
So like it doesn't look like normal. It's just like gooey.
It is. It's gooey. What color is it?
Like dark.
Brand new baby is dark.
I'm talking about brand new.
Brand new baby that has meconium. It's really dark. It's blackish.
Yeah.
Yeah. But you give it a couple weeks, and then, like, not even that, a week plus. Good for you changing a newborn's diaper. I'm talking an infant. This guy here, he wasn't just born. His poo isn't black. His poo is the color of the Nobel Prize that I put down there. It's this weird orange. Oh, by the way, to get the apple, you just have to tell me which is lactose and which is allolactose. If you want this apple. Brave, please. Give it a shot. Yeah, Nick is like, I'll take that apple.
Want that apple?
We'll get
Yeah,
it.
man. Take a second shot at it. So yeah, that's why I have the little orange icon down there on his booty. Why? What's going on? Well, it turns out that infants get the bulk of their vitamin B. Their vitamin B comes from the bacteria in their gut. The E. coli absorb lactose that the infant is eating, but it's an evolutionary advantage for humans because one of the byproducts, they produce a whole lot of B vitamins, like thiamine, folic acid, which is so important for neural development, niacin, the whole complex of B vitamins get produced. That is why baby poop is orange. Does anybody drank one of those like Adwala blueberry things or whatever, blueberry juice? What does your urine look like maybe a few hours later? That color, really dark orange. Or if you take a multivitamin, or you eat a bowl of Total that's like packed with all those vitamins, you're actually just pissing it out, right? When you eat a multivitamin, most of that is just water soluble being excreted. But it is all of these B vitamins that are being produced by the bacteria in your gut that are so beneficial for babies. This is why the lac operon is important. This is why mother's milk is important. Okay, is that too much of a soapbox? Please consider it. Yeah.
So can you just like say that like that little short piece again when you said like how the B vitamins are produced? You said
Yeah.
E. coli feeds on the what?
Lactose. Yeah, they feed on the lactose using the lac operon, which gets turned on by the allolactose. And the trade-off is babies get a lot of B vitamins in their colon from the E. coli. All right? Yeah.
So you're talking about kind of like being on like a soapbox or whatever. So like I guess like what happens like so then I would assume there's like I guess what are the consequences of like feeding your child formula? They just like don't get these vitamins like what?
Yeah, I was formula fed because my generation of people, the Generation X or whatever, at that time when we were children, it was culturally, our parents the baby boomers, they grew up in a time after the war when convenience was prime, right? And and technology and science could solve all of your problems, and progress came in a can, kind of thing. A lot of people started using formula rather than breastfeeding. In terms of actual outcomes, I'm actually not aware of, and I'm sure there are studies, but I can't quote you research. Maybe that's a slide I need to chuck in here to like take the story to the next level, because you're asking the logical next question. Is there, with access to formula, first of all, formula is easy to pack with B vitamins. All right, so when a baby is eating formula, their poo still looks yellow, but that is because there are B vitamins that are in the formula. They recognize that is an important nutrient that babies need, so they do stick that stuff in there. I think so. In terms of like cognitive development, it's probably unlikely that you would see an effect, a different effect between the two of them. In terms of the overall effect take eating formula and also having a C-section where you're not being inoculated with by the perineum of the mother, it's probably much more easy to tease out a difference in terms of effects on the immune system of those infants and other effects on the microbiome between an individual that was breastfed and vaginally birthed versus a C-section or an infant that was fed on formula. Another thing about formula is, I do know this is true, that infants that eat formula for a similar, when you compare quantities of calories consumed of breast milk versus formula, infants on formula tend to put on weight faster, which is not necessarily a good thing. It's not like you want the baby to be slim or something like that, but it means that they are assimilating more of the lactose, right? So if you give 40 grams of lactose via breast milk and then 40 grams of lactose via formula, more of the lactose in the formula is going to be assimilated, whereas in the child with that had been breastfed, less will be. So you'll have a larger spike in blood sugar, and the effects on basal metabolism and long-term metabolism and long-term health. I wouldn't be surprised if they have identified those correlations. I'm floundering because I don't have numbers to show you and tell you, because you're asking a very important, real, but data-driven question that I don't have the data to give you. Yeah.
So, do you know if like babies that are born by C-section, like,
yeah,
can the bacteria that they are exposed to be synthesized?
So there's something called vaginal swabs that they do, or perineal swabs that are becoming more common. So baby goes through C-section, the obstetrician takes a vaginal or perineal swab, and then she would inoculate the baby around the nose and mouth.
That's the same effect?
Yeah, I don't know. Same effect, but it has an effect apparently. Yeah. Okay, let's see if I can get to the end of lactose here. Okay. So, yeah, here's Allolactose crystal structure, and the only reason I'm putting this up here is, first of all, to help you appreciate the difference between lactose as a beta 1,4 versus allolactose as a beta 1,6. They both have gal and gluc, but they're just linked a little bit differently. The diagram I'm showing you is the crystal structure of them. I will say that that's a crystal structure I generated myself, which I'm kind of proud of, because that 1,6 linkage in allolactose, because there are more links in the chain, that's a really flexible linkage. The structure of allolactose is much more variable than the structure of lactose. And because of that, it's actually harder to hydrolyze, very hard to synthesize, and nearly impossible to crystallize. But I was the first and only to do it. Only one other 1,6 linkage got crystallized before, and that was Gentobios. So it took me years to do that. That's the only time I'm going to brag in class. So that's it. I've been waiting for years to talk about allolactose, and that's the first chance I've ever had. There it is. Okay, so that's it. That's class. We'll start here by expanding our concept of the function of carbohydrates next time. Any questions? See you on Friday.